colleagues, and friends. It's an honor to welcome you all to the 6th International Conference on Agricultural, Biological, and Life Sciences. I'm deeply thankful for your participation here, which symbolizes our common commitment to promoting knowledge, fostering innovation, and addressing today's urgent challenges. This conference is more than just a gathering of minds. It's an opportunity to cooperation, learning, and inspiration. Every great meeting like this is the result of a team who worked really hard to make it happen. Now, I invite you to stand for a moment of silence and then followed by the Turkish national anthem. İstiklal Marşı. We are now watching the promotional video for Trakya University.
Edirne, Osmanlı Devleti'ne 92 yıl boyunca başkentlik yapmış, köklü tarihi ve zengin kültürel mirasıyla her zaman dikkat çeken özel bir şehir olmuştur. Türkiye'nin en batısında yer alan bu güzel şehir, Doğu ve Batı Avrupa kültürleri arasında bir köprü görevi üstlenmiş, asırlardır farklı medeniyetlerin buluşma noktası haline gelmiştir. Şehrin zengin tarihi dokusu insanları adeta bir zaman yolculuğuna çıkarır. Camileri, köprüleri, kervansarayları, hanları, hamamları ve medreseleriyle şehir adeta açık hava müzesi görünümündedir. Sadece günümüzde değil, tarih boyunca da önemli eğitim kurumlarını içinde barındıran Edirne'mizin üniversite geçmişini 15. yüzyıla kadar dayandırmak mümkündür. Asırlar önce Sultan II. Bayezid tarafından yaptırılan tıp medresesi aynı zamanda üniversitemizin tarihsel geçmişini simgeler. Bu kadim şehir yalnızca tarihi ve kültürel zenginlikleriyle değil, Aynı zamanda çağdaş yaşamın her alanında gelişen dinamik yapısıyla da dikkat çeker. Edirne'nin stratejik konumu, doğayla iç içe geçmiş yapısı sadece geçmişte değil, gelecekle de sıkı sıkıya bağlı olmasını sağlar. Bu güzel ve özel şehirde 40 yılı aşkın bir süredir eğitim veren Trakya Üniversitesi, her geçen yıl kendisini biraz daha ileri taşırken, bu kentin bilimsel ve kültürel gelişiminin yanı sıra, ekonomisinden sosyal hayatına kadar her alanda gelişip kalkınmasında önemli bir rol oynar. Trakya Üniversitesi, 40 bini aşkın öğrencisi, alanında uzman akademisyenleri, dinamik idari kadrosu ve modern kampüs imkanlarıyla sadece Edirne'nin değil, tüm bölgenin gelişimine katkı sağlayan bir bilim yuvasıdır. 14 fakülte, 5 enstitü, 5 yüksekokul, 10 meslek yüksekokuluyla her geçen yıl büyümesini ve gelişmesini sürdürmektedir. Trakya Üniversitesi, geçmişten aldığı mirası geleceğe taşırken sürekli olarak yenilikçi yaklaşımlar geliştiren, bilimin ışığında ilerleyen bir kurum olmanın gururunu yaşamaktadır. Edirne'nin zengin kültürel ve sanatsal mirasını geleceğe taşıma konusunda Trakya Üniversitesi, kültürel değerlerin korunması ve geliştirilmesi konusunda geleceğe miras bırakacak projelere imza atmaktadır. Her yıl düzenlenen konserler, tiyatro gösterileri, sergiler ve diğer etkinliklerle sadece üniversite camiasını değil, tüm Edirne'yi kültür ve sanatla buluşturmaktadır. Üniversitemiz öğrencilere sunduğu ileri düzey laboratuvarlar, araştırma merkezleri ve dijital altyapı ile bilimsel araştırmaların öncüsü olmayı hedeflemektedir. Üniversitemiz akademik kadrosunun niteliği, müfredatının çağdaşlığı ve sunduğu öğrenci merkezli yaklaşımıyla her zaman kaliteli eğitimin adresi olmuştur. Üniversitemizde yürütülmesi planlanan bilimsel projelerin hem Türkiye'de hem de uluslararası arenada dikkat çekecek düzeyde olması bu üniversitenin önemli hedefleri arasındadır. Trakya Üniversitesi sadece ülkemizin değil, tüm Balkanlarda tanınan ve tercih edilen bir eğitim kurumu olma vizyonunu kararlılıkla sürdürmektedir. Balkan coğrafyasında köklü tarihi ve kültürel bağlara sahip olan üniversitemiz, bu bölgede eğitim görmek isteyen öğrenciler için bir cazibe merkezi olmuştur. Balkanlardan gelen öğrenciler, Trakya Üniversitesi'nde kendilerini evlerinde hissederken, bu bölgelerle olan akademik ve kültürel işbirliği projeleri, üniversitemizi uluslararası arenada da önemli bir konuma taşımaktadır. Gelecek nesillerin ihtiyaçlarına yanıt verecek şekilde kendini sürekli geliştiren Trakya Üniversitesi, Türkiye'nin ve Balkanların bilimsel ve kültürel geleceğini şekillendiren öncü bir kurum olarak varlığını sürdürmeye kararlıdır. Öğrencilerimizi bu köklü geçmiş ve parlak gelecek arasında köprü kurarak dünya sahnesinde etkin bireyleri yetiştirme kararlılığımız bundan böyle de devam edecektir. Şehri ve bölgesiyle daha bütünleşmiş, daha çok katkı sunan, daha güzel ve daha başarılı projelerle üniversitemizi daha ileri taşıma sevdası ve kararlılığıyla yeni bir döneme merhaba.
We will now listen to a short music recital from Trakya University State Conservatory Brass Band Group. Enjoy. I would like to thank you for this enjoyable music recital. And now, I would like to invite our esteemed organizer, Professor Yalçın Kaya, who has brought this great event to life with his dedication and hard work. Yalçın Kaya.
Good morning, everyone. Dear our rector, vice rector, dear colleagues, dear friends, dear our students, I would like to thank all of you. We are proud of you in the 10 years anniversary. We started this university in 2014. We have our guest year. There was a 2014 in the Agriculture Balkan Congress. So 10 years is our department, as our university, over 20 international organizations. So we are so proud of that, and to be together. A very, very big event. We started over 700 people in the participant in 2014. This is the same effort, the inspiration we are continuing. And again, in this year, we are organizing sixth agricultural biology conference. That this one, Agbiol, there's a sixth one, is 6 years, at least before the pandemic, just we make a break. So we are thank you, all of interest. In this year, there is a record participation from 55 countries all over the world, over 800 paper, and then there is about 2,000 authors we have. So we are thank you, very great interest. In this year, we organizing is the hybrid conference. Also, we are there is a live now in the Ortaköy University, and there's the YouTube. Also, we are broadcasting all of them. And again, there was our conference combining all the science and in the very nice environment, and our big opportunity for the Ortaköy University. You are all of the welcome you. And then we will be together until Friday. Of course, there is a post-conference tour, is the Cappadocia and also Balkan tour as well. So there is in the lunch will be available. We are calling Edirne Ciğer. There is a famous for the Edirne dish. So we will test for you. And then tomorrow also there is a Balkan in the Elbasan Tava in the lunch. We will be together, and Edirne Köfte also on Friday. So I hope you will enjoy our dishes, because Edirne, very historical place, and then also very nice dishes, and I hope you will enjoy our stay. And tomorrow evening we will be in the gala dinner, also in the Maritsa Riverside, is the Ortaköy University social place, restaurant. So bus will be available in the tomorrow evening at 6 o'clock here. So I don't want to take much time. I would like to thank again all of you, is our rector and our vice rector, also colleague, our invited speaker, our sponsor, all of you. Thank you all the participants. I really enjoy that. You give me the motivation to organize very, very nice event and continuously. So I hope, because Edirne is a very nice place for all of you, I think, to meet. I hope we can continue together. And then there is this is the nice, very big event as a record participation. Like every year, we are increasing the participation. I would like to thank again for your coming. I hope you will enjoy here is our university. Not only for of course, there's a science. In this environment, we have a good friendship, collaborationship. I hope we can collaborate in the future together. Thank you again. You are welcome.
I would like to thank you to Professor Kaya for his priceless speech. And then, it's with great pleasure that I invite Professor Mustafa Hatipler to the stage to share a few words with us. Mustafa Hatipler.
Çok değerli rektör arkadaşım, çok değerli rektör yardımcım, kıymetli bilim adamları, çok değerli misafirler, üniversitemizin çok kıymetli dekanları, enstitü müdürleri, okul müdürleri, Trakya Üniversitesi ve Uluslararası Araştırmacılar Derneği tarafından ortaklaşa düzenlenen Uluslararası Tarım, Biyoloji ve Yaşam Bilimleri Konferansı'na hoş geldiniz, şeref verdiniz.
Dear rectors, vice rectors, all the participants, professors, students, and deans, I welcome you all for the International Agricultural, Biological, and Life Sciences Conference, jointly organized by Trakya University and the International Researchers Association. We are honored by your presence.
Bu kısmı çevirmeyelim. Az önce çok güzel bir konser dinlediniz. Konserdeki en güzel parça Aşık Veysel'in parçasıydı. Gerçekten de uzun ince bir yoldayız. Hep birlikte yürüyoruz. Bu kısım çeviri dışında kalsın. Evet, bölgede tarımın merkezi olan şehrimiz Edirne'de böyle nitelikli ve yoğun katılımlı bir organizasyona ev sahipliği yapmaktan mutluluk duyuyoruz.
We are delighted to host such a prestigious and well-attended event in Edirne, a city that stands at the heart of agriculture in the region.
19. yüzyıldan bu yana yabancı birçok yaşam bilimci ve botanikçinin ilgisini çeken bölgemizde 1950 yıllardan itibaren Türk botanikçiler ve yaşam bilimciler tarafından yapılan çalışmalar neticesinde çok sayıda bitki örneği toplanmış ve floraya yeni türler de eklenmiştir. 300'den fazla endemik bitki türüne ev sahipliği yapan Trakya ve Edirne yaklaşık 50 kadar da türün dünyadaki en zengin popülasyonuna da ev sahipliği yapmaktadır.
Since the 19th century, our region has attracted the attention of many foreign life scientists and botanists. Since the 1950s, numerous plant samples have been collected and new species have been added to the flora thanks to the work of Turkish botanists and life scientists. Treysan Edirne, çok özür dilerim.
Devam et.
Home to over 300 endemic plant species, also hosts the world's richest population of about 50 species.
Böylesi farklı ve aynı zamanda endemik türleri de bünyesinde barındıran şehrimiz ve bölgemiz için önemli konuların ele alınacağı konferansımıza rekor düzeyde bir katılımın gerçekleştiğini görüyoruz, müşahede ediyoruz. Bilindiği gibi bu konferansların birincisi 2018 yılında, ikincisi 2020, üçüncüsü 2021, dördüncüsü 2022 ve beşincisi de 2023 yılında üniversitemiz ev sahipliğinde düzenlenmiştir. Alanın en prestijli ve en yoğun katılımlı konferansı olması da bu anlamda bizim için bir gurur vesilesidir.
We are proud to celebrate this conference where key topics concerning our city and region, which host a variety of unique and endemic species, will be discussed. I am pleased to see such a high level of participation. First held by our university in 2018, followed by editions in 2020, 2021, 2022, and 2023, this conference has become the most prestigious and widely attended event in its field, and we are very proud of that.
Bu yıl da rekor bir katılımla gerçekleşen konferansımızda alanının uzmanı davetli konuşmacılarımız ve dünyanın 55 farklı ülkesinden gelen yaklaşık 2000 araştırmacı ve bilim insanının sunacağı 800 aşkın bildirinin yanı sıra poster sunumları da gerçekleşecektir.
This year's conference has also set a record with more than 800 presentations by our distinguished invited speakers and around 2,000 researchers and scientists from 55 different countries worldwide, alongside poster presentations.
Bu kadar kapsamlı, nicelik ve nitelik açısından oldukça zengin bir konferans, üniversitemiz çatısı altında gerçekleştirilmesinde göstermiş olduğu emek, gayret, her türlü takdirin üzerinde olan ve arkadaşım olmasıyla, dostum olmasıyla onur duyduğum Çok sevgili Profesör Doktor Yalçın Kaya hocamıza huzurunuzda ve onun şahsında bütün çalışma arkadaşlarına ayrı ayrı teşekkür ediyorum. Sevgilerimi ve saygılarımı sunuyorum.
I would like to extend my heartfelt thanks to my dear friend, Professor Dr. Yalçın Kaya, and his colleagues for their tireless efforts in organizing such a significant conference in terms of both quantity and quality under the auspices of our university.
Konferansımız hayırlı sonuçlar getirsin inşallah.
I hope this conference goes very well for all of us.
We would like to thank to Mustafa Hatipler for his priceless speech. I think we can wait for Yalçın Kaya. We now invite Oğuzhan Erdem.
Okay. Good morning to everyone. Dear Vice Rector, Deans, my colleagues, and all guests, you are welcome to opening session of this 6th International Agricultural, Biological, and Life Science Conference. My name is Oğuzhan Erdem from Trakya University Electrical and Electronics Engineering Department. I will be chairing this session. Before we get started, let me give information about this session. We will have 3 talks, and I think we have tight schedule, so each speaker will have approximately 25 minutes for their presentation. At the end of this session, we will accept questions from the audience. So if you have any question about the presentations, please wait until after all the presentations are over. So our first invited speaker, Professor Fikrettin Şahin from Yeditepe University. He's going to present characterization and applications of plant exosomes in regenerative medicine. Before inviting the professor, let me read his short biography first. Professor Şahin was born in Erzurum in 1968. He graduated from Atatürk University, Faculty of Agriculture and Department of Plant Protection. Professor Şahin completed his Master of Science and PhD education at the Ohio State University, the Department of Plant Pathology from the College of Food, Agricultural and Environmental Sciences in USA. In between 1997 and 1998, he worked as a postdoctoral researcher in clinical microbiology at the Western Ontario University. He has worked at the Atatürk University, Turkey for 5 years, starting from 1999 to 2003, until he moved to Faculty of Engineering at Yeditepe University. Mr. Şahin is professor in the Department of Genetics and Bioengineering at Yeditepe University, where he has been serving as a faculty member since 2003. Professor Şahin's research focuses on molecular characterization of plant, soil, food, and animal or human-associated microorganism biological properties of molecules from medicinal plants, stem cell, and cancer genetics. He is the author of about 500 scientific papers, 500 abstracts, 40 book chapters, and 7 books, and he has 245 granted patents and more than 300 patent applications. He has been awarded by a number of national, international institutions due to scientific contributions he has done throughout his academic life. Professor Şahin currently serves as the chairman of Genetics and Bioengineering, director of Biotechnology Institute, coordinator of Research and Development and Analysis Central Laboratories. Professor has been honored as the principal member of the Turkish Academy of Science in 2012, the TÜBA Council member between 2013 and 2019, executive committee member and presidency of the Turkish Health Institute. Yes. Okay, so here you are, Professor. We are waiting your presentation. We are waiting your slides.
Hello, everyone. Good morning. Before starting my presentation, I would like to thank to the organization committee members of this conference, especially Professor Dr. Yalçın Kaya, who invited us to come together here in this great conference and share and exchange our knowledge and experience to each other. Today, I'm going to talk about plant exosome used in regenerative medicine. As you know, the regenerative medicine is an emerging and very important discipline in order to study and develop novel and new therapeutics to repair and regenerate damaged and diseased organs. Today, there are some alternatives available in this area used as therapeutic products, such as stem cell, gene therapy products, biomaterials, engineering tissue, and biologically active compounds. Extracellular vesicles derived from different sources are one of the very important sources for regenerative medicines. And extracellular vesicles defined actually as lipid bilayer vesicles secreted by all known type of the cells from different biological systems into the extracellular space. And the matrix or content of the extracellular vesicle generally different based on the secreting cells. And recent studies showing that different cells or the same cells under the different physiological condition, they may have completely different matrix. And we know that the cells can produce more than one type of the extracellular vesicles. And so far, the study is classifying extracellular vesicles into the 3 different types: microvesicles, exosomes, and apoptotic bodies. They are generally differentiated based on their biogenesis, releasing pathways, size, content, and biological function. And microvesicles, actually, the extracellular vesicles formed by budding of extracellular membrane, and they are generally larger than the other extracellular vesicles, such as exosomes and apoptotic vesicles. And exosomes are nano-sized extracellular vesicles, and their size in between the 30 to 100 nanometers, generally less than 200 nanometers, and produce inside the cells from the multivesicular bodies and secreted into the extracellular space through the exocytosis. Apoptotic vesicles are the extracellular vesicles produced by apoptotic cells. When we come to the exosomes, exosomes are the most favorable extracellular vesicles used nowadays in regenerative medicine, and as a scientist, we are hoping that in the future, they are going to be the most important sources for biotechnological drug formulations. First time in 1986, Drs. Trumps and Johnston identified the exosomes. They isolate the exosome from the culture supernatant of the red blood cells in vitro. And later on, all the other studies show that exosome, yes, is lipid bilayer nanovesicles produced by different type of the cells in different biological systems and secreted into the extracellular space in order to change biological function and physiology of the other cells. So that means the main role of the exosome in different biological systems is basically intra and generally intercellular communication between the cells. So far, yes, exosomes isolated, purified, and characterized and studied from different sources, but exosome from the mammalian system very well studied, and we know that the content very well. And the previous studies is showing that exosome abundantly found or present in all different type of the body fluid of the mammalian system, including the salvia, blood, urine, breast milk, and etc. So when we look at the exosomes today, as a scientist, we are working and using in the regenerative medicine, there are three important sources. Animal-derived exosome, plant-derived exosomes, and especially engineered exosome. Engineered exosome means the modified exosome. Basically, this modification can happen through the genetical or biological modification of the proteins on the surface of the exosome, or chemical modification, and that chemical modification can happen basically uploading or loading the therapeutics into the exosomes and then delivering them to the specific tissue or organs in biological systems. So that means we are using exosome, engineered exosome for targeted drug delivery or therapy. We said that mammalian exosomes studied very well, and we know that the content very well so far. We know that the mammalian exosome can include the lipid, protein, nucleic acid, and many secondary metabolites. In the case of the lipid, this vesicles is basically enriched in terms of the cholesterol, sphingolipid, fatty acid, glycerolipids, sterolipids, and steroids. For the proteins, general exosome contains surface receptor protein, enzymatic protein, some structural protein, what we call them cytoskeleton proteins, transmembrane proteins, cytosolic proteins, and in their functional form. For the nucleic acid, exosome include the DNA fragments or different type of the RNAs like miRNA, mRNA, or non-circular RNAs, in addition to some secondary metabolite, such as carboxylic acid, amino acid, vitamins, sugar, or cyclic alcohols, and many other secondary metabolites. When we come to the plant exosomes, we call them plant exosome, since it's not very well characterized so far. Generally, we are calling them the plant extracellular vesicles. Those are produced in response to the various biological or non-biological stress factors, including pathological infections, such as bacterial infection, viral infection, fungal infections, or some abiotic factors, stress factors, such as drought, salt stress, cold stress, flooding stress, and many other environmental factors. And we know that plant exosome also produce inside the plant cells in the same mechanism as the mammalian exosome, because they are also generated from the multivesicle bodies inside the plant cells and secreted into the extracellular space. And their main role in the plant biosystem is intercellular communication between the cells. And the recent study is showing that plant extracellular vesicles can be isolated from different parts of the plants, including the juice, leaves, stems, root, seed, or dried plant materials. And they can be characterized by their density, size, origin, and/or biological composition. Plant extracellular vesicles also, just like the mammalian exosome, they have nucleic acid, DNA, RNAs, proteins, lipid, and secondary metabolites. But, of course, biological composition or molecular content of the plant exosome completely different than the mammalian exosomes. This slide is basically showing that when we look at all different biological systems, the roles and biological function of the exosome used now by the scientists working in this field for transfer of the genetic information, cellular homeostasis, immunomodulation or immunoregulation, intra and intercellular communication between the cells, autophagy, control of the infection diseases, drug or gene delivery vehicles, diagnostic markers, and especially regenerative medicine. And scientists in our group at Yeditepe University, mainly their work is focusing in this area. So what we are doing at Yeditepe University, about 10 years ago, some scientists in my group, they came to me and they said, well, there is a new trend in the world, especially in the mammalian system. There is a specific vesicles containing so many bioactive components or the molecules they call the exosome, and this exosome is the basically future of the next generation drugs. There are a lot of studies in the field of the mammalian system, but not the other biosystem, especially for the plant. Why don't you involve in this area, establish labs, and find out that the scientists come together and work in this field? And I checked and find out, yes, this area is really interesting. So I decided to establish a well-equipped labs. That is the first labs in Turkey. We call them exosome lab, exosome isolation, purification, and characterization labs. And then scientists, including the graduate students, postdocs, researchers, faculty members, technical persons, all come together, and we make a huge team to work in the field of the exosome. What was the main focus of our studies in the exosome? Basically, isolation and characterization of exosome from different sources, not only from mammalian systems, but also plant, parasites, and especially microorganisms. And some scientists and researchers in my group, they would like to basically study the regenerative properties of the plant-derived exosomes by evaluating their effect on cell proliferation, cell migration, cell differentiation, angiogenesis, nerve regeneration, immune regulation, antioxidant, antimicrobial activities, and anti-cancer activities tested in vitro and in vivo studies, especially develop new formulation. And this new formulation is exosome-based formulation that we can test and use in the field of this cell-to-cell signaling, cell-free therapeutics, cancer treatment, and drug delivery systems. When we start to work with the plant exosome, we found out there are some gaps in literature regarding to the plant exosome. In the field of the isolation and purification protocols, not very well established. No biological markers available or known for the plant exosome characterization, or content of the plant-derived exosome not very well known and studied. Only a few omic data available for specific for the certain plant species, and also bioactive content of the plant-derived exosome and their biological role in regenerative medicine not very well studied. So that's why in our studies, we decided to first look at or evaluate the currently available protocol used for isolation and characterization of the plant-derived exosome or exosome derived from different sources, and plus develop a new protocol to isolate and identify and characterize the plant-derived exosome with high quality and quantity. And if you have this data, then you can do the comparative study to look at the similarities and differences between the exosome derived from different sources, and then you can, of course, develop the potential biomarkers for plant exosome, and you can find especially omic data to identify and characterize plant exosome that you are interested in. In this study, when we start in our work, we look at the currently available protocol for isolation of the plant exosome. We found out there are especially centrifuge-based protocol, what we call them sucrose cushion protocols, and some kit-based protocols. Whatever the protocol you use at that time, when you isolate the plant exosome contaminated with either some chemicals coming from the reagent used in protocol, and especially endosome contamination in plant exosome. Where the endosome is coming, basically? When we look at the protocol, we found out that very early stage of the isolation protocols, we are using plant samples. That plant sample either coming from the field or greenhouses, either fresh samples or dried samples. What we do at that time, we generally use the mechanical protocols to scratch and crush the plant samples. And then, of course, you are damaging the plant cells. And then when you use the protocol, you isolate the exosome. Of course, your exosome is contaminated with the endosome coming from the mechanically damaged plant cells. In order to avoid this problem, to get rid of this problem, we decided to develop new protocols specific for different plant species to grow the plant in suspension culture. When you grow the plant in suspension culture, you can easily separate these cells from the supernatant. An exosome, of course, inside the supernatant. And you can use the supernatant either using the currently available protocol to isolate the exosomes or the protocol that we develop in our group. That protocol, we call them PEG-DEXTAN two-phase exosome isolation protocol. This protocol is basically we developed to isolate and purify the exosome not only from the plant samples, but from different sources, including the parasite culture, serum, mammalian culture, microorganism cultures. And then we internationally patented this protocol. After that, we publish the data or the protocol in scientific report journals in 2019. Now that the protocol available, we are using in our studies. If anybody here interested in working with the plant exosome, you can download this protocol and use without paying, actually. And this slide is basically showing, of course, when you isolate and purify the exosome, the next step is the characterization. And this slide is showing that one of our papers we published a few weeks ago in Biofactor Journal. In this study, we grow the stevia plant in suspension culture. After using the isolation and purification protocol that I have mentioned, and then for the characterization, we use nanosite, or what we call them nanoparticle tracking analysis, in order to look at the size and size distribution, and also quality and quantity of the exosome we have isolated. After that, we use different imaging systems. Atomic force microscopy, scanning electron microscopy, and transmission electron microscopy to analyze the physical properties of the exosome. After testing, doing all those characterization, then next study we have completed omic studies. We evaluate the omic profile of the exosome. This omic study is basically including the lipodomic, as you see the data here, proteomic, transcriptomic, and metabolomic. Once you have all those omic data, what you can do, basically, you know the matrix of the exosome. This exosome may be derived from different plant species, right, or different sources. So then, easily you can do comparative study to look at the similarities and differences between the exosome, and easily you can find out or determine some potential biomarkers you can use for exosome, plant exosome, and at the same time, some omic profile is easily, unique omic profile you can easily use for identification and characterization of the plant exosome that you are interested in. That's what we have done so far. And this slide is basically showing that when we analyze the protein profile of the Nicotiana tabacum exosomes, we find out that the 8 proteins I have listed in this table showing that is the potential biomarkers for plant exosomes. I'm not going to give the names, but if you are interested in, I can share the data. And after that, well, we decided to study regenerative properties of the plant-derived exosome. Of course, now you have the product available, you already isolated, and you have purified, and you have characterized, and you have the plant exosome and the exosome from the microorganism, exosome from the mammalian systems from different sources. And then now you can look at the regenerative properties, especially since I'm going to give the more detailed information about the plant-derived exosome. So here I'm sharing just plant-derived exosomes result. One of the important areas in regenerative medicine is the cartilage regeneration or cartilage regeneration. As you know, the cartilage is a connective tissue found primarily in the joint of the mammalian system. And generally, the cartilage, once the cartilage damage happens, and the cartilage has very limited capacity for self-regeneration. And many researchers demonstrated that exosome derived from different sources, including the plant exosome, also has critical role for regeneration of the cartilage and bone. Oops. What happened? Okay, thank you. In our studies, first we have selected 5 different plant species to do the in vitro studies first, looking at the cartilage regeneration. And those plant species, beet, lemon, radish, tomato, and wheat. We have isolated and purified the exosome from those 5 different plant species, and then we decided to look at the effect of the plant-derived exosome on cell viability and cell toxicity on adipose-derived mesenchymal stem cells. So we have tested different concentration in vitro in MTSSA, and our result show that none of the concentration we have tested, exosome concentration we have tested from 5 different plant species has toxic effect on mesenchymal stem cell. When you look at also those figures, you can see that even the increasing concentration of the exosome increase also cell migration and cell proliferation in MTSSA. And then in the second step, we decided to look at the effect of the plant-derived exosomes on chondrogenic differentiation of adipose-derived mesenchymal stem cells in vitro condition. And this slide is showing that microscopic imaging in alkaline blue staining assay, and our results show that except lemon exosomes, all the exosomes we have derived from the wheat, radish, beet, and tomato trigger the chondrogenic differentiation in vitro condition. Then we would like to also look at and evaluate the expression level of the chondrogenesis-related genes in adipose-derived mesenchymal stem cells treated with the exosome from different plant species with different concentration and in vitro condition. And our results show that our result, basically real-time PCR result, confirming the alkaline blue staining assay that except the lemon, wheat, radish, tomato, and beet exosome increased the upregulation of all chondrogenesis-related genes in adipose-derived mesenchymal stem cells after the treatment. Then, after completing all those in vitro studies, we would like to see that plant exosome is whether it is working in vivo system, in preclinical system or not. So we want to look at also the effect of the plant-derived exosome treatment on regeneration of the cartilage damage in a rat model. We use, in this case, in the preclinical study, radish exosome and Sukash exosome. And our result, histopathological study result, showing here in this slide that radish exosome treatment increase healing or regeneration of the cartilage damage in rat model, but not Sukash one compared to control. Another important area in regenerative medicine is the wound healing. We want to look at also the plant exosome as a cell-free therapeutics, whether it's working in the wound healing or not. You know, the wounds are known to be the injuries on the skin or other body tissues caused by physical, chemical, and biological insults. And recent studies showing that exosome derived from the mammalian systems or plant has great potential to enhance the wound healing. So in our studies, in this case, we decided to use the wheat-derived exosome to look at the effect on the wound healing in vitro condition. We established two different in vitro studies. One of them is the in vitro tube formation assay on matrigel to look at the endothelial differentiation of mesenchymal stem cell. And the second one, we use the in vitro scratch assay, and then we would like to look at the effect of the wheat-derived exosome on cell migration and cell proliferation on three different cell lines. We used HUEC, that's endothelial cell, and SDF, and HACAS cell line. And our result in vitro show that wheat-derived exosome enhance endothelial differentiation or angiogenesis, and also increase the cell migration and cell proliferation in vitro test we have completed. And our result already, in vitro result in this study, we published in Applied Biochemistry and Biotechnology Journal in 2019. After completing in vitro study, again, we would like to see our result in preclinical conditions and in animal model. We use a rat model, and which evaluate wheat-derived exosome on the wound healing, and our results show that wheat-derived exosome promoted the wound healing by decreasing healing time and inflammation in a rat model, based on the histopathological test result. Another area in regenerative medicine, of course, cancer treatment is always important in regenerative medicine, and we would like to see how good plant-derived exosome used as therapeutic in cancer treatment. So we conduct two different in vitro studies. One of them is the annexin 5 apoptosis-based assay to investigate the anti-cancer properties of, in this case, garlic-derived exosome. We have tested on three different cancer cell lines. One of them is the small cell lung cancer, second is the colon cancer, and third one is the breast cancer cell lines. And our result, and here in this study, we use HUEC as the healthy cell lines, and our result, annexin 5 assay results show that optimized concentration of the exosome here in this case is the 75 microgram per milliliter, and garlic exosome triggered more than 70% apoptosis in all three different cell lines we have tested. And this concentration, we didn't see any cytotoxic effect on healthy cell lines. The second in vitro study we have completed to look at the apoptotic and apoptotic gene expression level of the cancer compared to the healthy cell lines after the garlic treatment in real-time PCR data, and our real-time PCR data also show that expression level of the apoptotic genes increased and anti-apoptotic genes decreased in all cancer cell lines we have tested after garlic exosome treatment. And this result actually is the first result in literature showing that garlic exosome has anti-cancer effect. Now we are doing some clinical studies and further studies. We are hoping that soon we will be doing also some clinical studies. We hope that soon we can develop a new formulation, garlic exosome formulation that we can use for treatment of the different type of the cancer in clinic, and we can develop a novel drug formulation in the future. And result of this study is already published in Scientific Report Journal a year ago. And finally, I would like to mention that we are using the plant exosome in also cosmetic industry. So far, one product we have developed and introduced to the Turkish market, it's called Again. That's the cosmetic product. That's a basically spray. And this spray is including the exosome from 3 different plant species, garlic, pomegranate, and pineapple. And that product, as I said, that product is introduced to the Turkish market 3 years ago. And we know that the effect, we have great effect. Whoever used, they are very happy, helping the hair growth, and reducing the hair loss, and giving capacity to the hair. Now we are developing an injectable form, and we can use in clinic also. And not only just this product, and we have many other products ready in the line that we are hoping that soon future we will be able to introduce to not only Turkish market, but the international market. That's all today I'm going to talk about. Before finishing my presentation, I would like to thank all the scientists in our exosome group, including the technical persons, graduate students, researchers, faculty members, or visiting scientists working with us. They have great contributions in our studies, and I would like to thank each one of them. I'm not able to list their names because they are too many, and I would like to thank you guys for your patience. Of course, I would like to take your question, but Chair told me that you cannot take the questions just directly after your presentation. You have to wait at the end of the session. So I'm stopping now here. I hope I didn't take so long time.
Okay. Thank you, Professor, for your great presentation. We are going to get questions at the end of this session. Okay, we come to the next presentation. Our second presenter, Professor Ahmet Uludağ from Çanakkale 18 Mart University. The title of his presentation, The Effect